quantum tunneling in enzymes. Inside your body right now, billions of chemical reactions are occurring that, according to classical physics, should be impossible. These reactions happen not because the laws of physics are being broken, but because they're being bypassed through a quantum mechanical phenomenon called tunneling. This process is particularly crucial in enzymes, the biological catalysts that make life possible. Enzymes are specialized proteins that accelerate chemical reactions in living organisms. Without these molecular machines, the reactions necessary for life would proceed too slowly to sustain biological functions. Your body contains thousands of different enzymes, each designed to facilitate specific chemical transformations. They're responsible for everything from breaking down the food you eat to replicating your DNA when cells divide. Enzymes increase reaction rates by factors of millions or even billions compared to uncatalyzed reactions. This acceleration allows processes that would naturally take years to occur in fractions of a second. Understanding how enzymes achieve this remarkable efficiency has puzzled scientists for decades. Chemical reactions require molecules to overcome energy barrier, like climbing a mountain before transforming into products. This barrier, the activation energy, determines reaction speed. Traditional enzyme theory explains that enzymes lower this barrier by holding reactants in optimal positions. However, this explanation only partially accounts for enzyme efficiency. Scientists consistently observed reactions occurring hundreds of times faster than classical models predicted, particularly for hydrogen atom transfers common in biological systems. The solution came quantum mechanics and the phenomenon of tunneling. In the quantum world, particles exist as probability waves rather than definite objects. This wave-like nature allows particles to occasionally pass through energy barriers instead of going over them. Imagine a shortcut through the mountain rather than climbing over it. Quantum tunneling creates these impossible pathways. Hydrogen atoms, being the lightest elements, exhibit the strongest tunneling effects. Their small mass gives them a more pronounced wave-like character, increasing their tunneling ability. This property becomes critical in many enzyme reactions involving hydrogen transfers. In 1989, scientists at MIT conducted a groundbreaking experiment with the enzyme alcohol dehydrogenase, which breaks down alcohol in your liver. They replaced normal hydrogen atoms with deuterium, a heavier hydrogen isotope. If classical physics alone governed the reaction, this substitution would cause only minor slowdowns. Instead, they observed reaction rates dropping dramatically by factors of 50 or more. This provided compelling evidence that quantum tunneling was essential to the enzyme's function, as the heavier deuterium atoms couldn't tunnel as effectively as hydrogen. DNA polymerase demonstrates this quantum advantage remarkably. This enzyme replicates DNA with an error rate of less than 1 in a billion. Like a quantum courier taking shortcuts through an impossibly congested city, hydrogen atoms tunnel between DNA bases, allowing the enzyme to check and correct potential errors with extraordinary speed and precision. Without tunneling, DNA replication would be too slow and error-prone to maintain genetic stability. Scientists have used advanced spectroscopic techniques to directly observe hydrogen tunneling in enzymatic reactions. These methods track atom positions with extraordinary precision, showing that hydrogen atoms don't follow classical paths, but instead tunnel through energy barriers. Enzymes haven't just passively adapted to quantum effects, they've evolved to optimize them. Their structures create environments that position donor and acceptor atoms at ideal distances for tunneling. Protein dynamics, subtle movements within enzyme structures, bring reactants into alignments that maximize tunneling probability. This fine-tuning suggests that quantum effects aren't incidental, but essential to enzyme function. Beyond DNA polymerase, quantum tunneling appears in enzymes involved in cellular respiration, photosynthesis, and various metabolic pathways. Soybean lipoxygenase shows some of the most dramatic tunneling effects ever measured in a biological system, accelerating hydrogen transfer rates by over 1,000-fold compared to classical predictions. Quantum tunneling contributes to both DNA mutation and repair. Occasionally, hydrogen atoms and DNA spontaneously shift positions through quantum effects, creating tautomeric forms, alternative arrangements of the same atoms that can lead to mismatches during replication. These quantum events contribute to the background mutation rate driving evolution. Repair enzymes then use tunneling themselves to identify and fix these changes, creating a balance between genetic stability and evolutionary variation. This quantum view transforms our understanding of life at a fundamental level. Biological systems have evolved to harness and control quantum effects. The precision of DNA replication, the efficiency of metabolic processes, and ultimately the stability of the genetic code all depend on quantum tunneling. The discovery that life depends on quantum effects raises profound questions about how such systems evolved. The enzyme structures facilitating quantum tunneling require precise atomic arrangements encoded in DNA, which itself relies on quantum tunneling. This creates a remarkable interdependence where quantum effects enable the very systems that have evolved to harness them. When considering life's origin, we must account for the emergence of structures optimized for quantum mechanical processes. This adds significant complexity to mathematical models calculating the probability of life arising through random processes. As we'll see next, when these quantum processes are incorporated into calculations about DNA's formation, the mathematical picture becomes considerably more intricate.